Well, I will talk you about the genetic code evolution as revealed by proteins and RNA. Well, first of all, I would like to thank to uh, Norcell's founder and chairman Sohanjita for allowing me for giving me this opportunity to presenting this talk to you. Well, as we saw with the Richard Gordon and Savio presentations, we have several scenarios for the origin of life, several hypotheses. We have on one side we have the clay world, the membrane world, and the thermal vent world, the metabolism first world, and the RNA world. Of course, those scenarios have many supporters, have many experiments to prove somehow. But well, nowadays, as Richard Gordon said, we have no any certainty about what what happened at the origin of life. For example, yes, we have a timeline uh, to uh, to to accommodate the steps in the origin of life. For example, we have the formation of Earth, we have then a hydrosphere, and we began with the prebiotic chemistry. Later on, we have the RNA world and the DNA, the first DNA protein life, and later on, we have the last universal common ancestor. Actually, as Richard Gordon says, for example, and also as Savio Torres suggested, we don't know exactly how and when the life started, but we can only delineate some scenarios for this transition from the prebiotic to the protobiotic life or stages of the matter. We have, for example, the RNA world that is followed by a ribonucleoprotein world, as Savio, for example, mentioned in his talk, and we have the last universal common ancestor, as I told you, but the characteristic of the LUCA is that it was a living organism whose genetic material was DNA. So we have two main approaches, two main types of approaches that have allow us to trying to 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 figure out which was the first characteristics for the first living organisms. On one side we have the bottom up approaches, and on the other side we have the top down approaches. And in the middle of the pathway, we could have the characteristics of the first living entity, supposedly. But we are focusing only on one type of model that is called the genes first. The genes first, the main exponent of this scenario of genes first is Manfred Eigen, who was worked on how the first ribonucleic acid chains configured. For example, Eigen said that the genetic code is not older than, but almost as old as our planet. The main contribution of Eigen was to state that the first genetic code was made of ribonucleic acid chains with an RNY pattern. What does mean RNY? The R means for purines, adenine or guanine. Y means for the pyrimidine, cytosine, or uracil, because we are talking about RNA, and the N stands for any base. We have so an schema of this RNA, 5 prime to 3 prime, and it follows the RNY pattern sequentially one after the other. Is a RNY pattern RNA with only RNY pattern chains. So, well, here is more detail about the Eigen and Schuster proposal about the first genetic code, because well, they gave three main uh, three main points about reinforcing the idea that the RNA, the first RNA code was based on RNY pattern. So we have that basically the ribonucleic acid chains we have are have the self-reverse complementary also made by RNY pattern. We have here, for example, one chain, 5 prime to 3 prime, and the other chain that is reverse complementary with the same characteristics. Well, we have then the first genetic code that is based on RNY pattern, and it is composed of 16 codons. We then have two extended codes that is formed by frame shift reading mistranslations, or the extended two that is formed by transversion in first or in third base. According with a work by Jose Marco in 2009, it exists an algebraic derivation that has biological equivalence for this dichotomic pathway, extended one and extended two, and this is the publication about this explanation. And as I told you, it has an algebraic derivation. Now, for example, we have in the right side of the screen, we have the entire triplets and the amino acids encoded by them. All the triplets that are part of the RNA world, according to Eigen's ideas, and the two extended genetic codes, the extended one and that contains the three stop codons and the extended two on the other side. As we can observe, the both extended genetic codes complement to each other to finally conform the standard genetic code. So we could position the three types of genetic codes in the timeline based on the Tomasek work about the RNA world, forming later the ribonucleoprotein world, and later on the LUCA. Yes, I read the chat about Ellen Hansma commented that it's important to distinguish the ribonucleoprotein world because yes, it is not about proteins totally formed since the beginning, but at the beginning the formation was more like to peptides, short peptides that were increasingly growing, as and in our model, we can distinguish this evolution of from peptides to proteins. So the work by Jose Marco says that the genomes with only RNY extended 1 or extended 2 triplets show a scaling variance with respect to the original genome, and it says it indicates an evolutionary conservatism. It is a fractal nature. What I mean with a fractal nature? Well, as imagine that you have the whole place of Shakespeare, of William Shakespeare, and we take, for example, some synopses, summaries, and very short and later on a very short summary about the Shakespeare plays. Well, it's the same with the genomes. We have, for example, the RNA genetic code, and later on the 2 extended genetic codes. But in all of those genetic codes, we can observe the same characteristics that that defines specific organisms. Of course, we have another proposals about also the evolution of genetic codes. For example, we have the supporters for a coevolutionary scenario that is based on the metabolism first scenarios at the molecular level. For example, the exponent of this coevolution scenario, we have Cephei Wong, Taylor, Davis, Di Giulio, Cardoso Guimaraes, but the directionality of this coevolution scenario for us is unclear. We have also the scenario about Ikehara that has modeled the first genetic code based on only on triplets GNC. But we can observe that GNC are indeed RNY pattern. We have also a genetic code evolution scenario based on the amino acid RNA ligases evolution. However, it was discovered that the several scenarios are mathematically equivalent to the RNY model evolution. We have, for example, two works about the unified model or the uniqueness of the standard genetic code, and there is modeled using algebraic derivations, and in all of those in these works, we can observe that for the RNY genetic code as the first genetic code, and the model by Rodin Ono and the LaRue models, all of them are mathematically equivalent. So we have, for example, we have that this algebraically, this pathway, this dichotomous pathway, starting with RNY pattern and the 2 extended genetic codes, is biologically plausible, and it is algebraically sounded. But what we did to prove, trying to prove with proteins this mathematically derived model. Well, we looked for the proteins or the peptides encoded by the first genetic code, by the RNY genetic code. and we obtained that the proteins were not proteins indeed, but the first peptides, we obtained the first peptides encoded by the first genetic code, by the genetic code proposed by Eigen. We have, for example, um, um, the we have the peptides encoded by the primeval genetic code, and we call it the Ur proteome. And the Ur proteome implies that the prebiotic amino acids that existed, for example, by protochemical or prebiotic chemistry, for example, glycine, valine, leucine, for example, those amino acids interacted with the RNA strands that were made of concatenated RNY triplets. And this caused that the ancestral peptides to be encoded produced by the 8 amino acids phenotype, implying thus the emergence of the first genetic code. So what we found is that the ancestral peptides, nor proteins, not in that stage, in that evolutionary stage, but ancestral peptides constitute mostly the binding site for nucleic acid chains, or the binding site for molecules with a nucleotide moiety, for example, for ATP, NAD, a flavin adenine dinucleotide, a coenzyme A, etc. Or the binding site for ions and other small molecules such as water or glycerol, or constitute the binding site for random peptides or amino acids in repeating motifs. But that small region can be considered the primordium of the current protein, uh, and possibly the primordium of related proteins. Therefore, it constitutes the Ur proteome. And given that those models are binding sites for molecules that could be considered as cofactors, we call them the cofactor stabilizing binding sites. So this collection of ancestral peptides, these ancestral modules, can be referred as the bindome. It was published in 2018 this paper was presented at a conference at ISOL, thanks to Dr. Sohanjita. And later on, we try to figure out using the same reasoning, but now we try to figure out the proteome encoded by the extended codes, extended 1 and extended 2. We use this set of organisms, and we found the same finding by using only one organism, and now by using 26 organisms, bacteria and archaea. We found that these proteins contains regions encoded by RNY triplets, and that regions are that regions are the cofactor stabilizing binding sites. We then found the proteins with regions encoded by the extended genetic codes. So the results are very similar with that we found in the first stage, and the proteins that contain portions encoded by each of those genetic codes are distributed in all cellular pathways, and the proteins related to RNA or DNA are more abundant. But we have to remind that those are only the portions. It doesn't mean that DNA proteins were existence before Luca at this stage of life, not necessarily. Well, we have also several distributed in several metabolic pathways, and some others are related with the transport of various molecules. Well, none of those extant proteins encoded by extended genetic codes are completely encoded by those extended genetic codes. But uh, not for all proteins. Some of them are almost completely formed before Luca, but not all of them. So, well, we have several um, coincidences with other works. For example, coinciding with the work by Sabio Farias, that found proteins encoded by proto-tRNAs concatenates, and also with the proteins encoded by the extended genetic codes are, for example, coincide with the proposals that the Wood-Junghal pathway was the first energetic pathway, according with a work by Rachel Weisz and William Martin, that proposed the Wood-Junghal pathway as the metabolism of LUCA. We found the proteins of that pathway, but for example, in that work, they don't find the key proteins, the key enzymes. So, for example, the acetyl coenzyme synthase or the ATP synthetase, and we found that proteins, that key proteins. But the Wood-Junghal pathway could be indeed the most ancient metabolism, probably. So, for example, we have here the three-dimensional reconstruction of those uh, peptides encoded by the RNY genetic code, or by the extended 1 genetic code in orange, or by extended 2 genetic code in purple. No, in blue. The purple is the original structure determined by crystallographic studies. So, for example, we have here a portion of a ribosomal protein, the S11 ribosomal protein, and we can trace this type of proteins to the RNY genetic code, to the primeval genetic code. We have also here the triose phosphate isomerase that has been proposed as one of the most ancestral proteins, ancestral enzymes, and we observe that we can reconstruct almost the whole protein by an encoding in any of both extended genetic codes. We have, as I told you, one of the key enzymes of the Wood-Junghal pathway. We found, for example, the subunit alpha of the acetyl coenzyme A synthase that is encoded by extended 1 and extended 2 genetic codes. Or, for example, we have the gamma subunit of this protein that is encoded that whose a small there is a very small portion of this protein even that can be traced to the primeval genetic code, and the extended genetic codes encoded almost the whole protein. Perhaps fully is not fully encoded by this genetic code, but almost completely encoded. So we gotten the evolution of proteome, from the primeval genetic code and the encodings into a standard genetic by two extended genetic codes. So we could predict the structures and the probable ligands of some of those proteins. And we think that our findings fill in part the gap in the understanding of evolution of proteins since the first form of life to the proteins of progeners, but before the emergence of LUCA. Later on, we consider the prebiotic RNA world scenario and how could have evolved. Uh, how could have evolved? Well, based on the proposal by Eigen and Schuster on the primitive translation system, and based on the algebraic derivation that I mentioned of the standard genetic code from the RNY code on their symmetry breaking model, and as we find the vestiges of the primitive genetic code in current genomes as critical scale invariants, then we propose that it's possible to trace also not only the proteome, but also the evolution of the RNAome, using again the codification in RNY, extended 1 and extended 2 triplets. So we it's just a reminding that what does mean the RNY code that is formed by 16 codons, the extended 1 genetic code that is a 48 codons, 32 plus the RNY, and the extended 2 also 48 codons, different from the extended 1 in some triplets. That is that also takes into account the RNY codons. So, what were the types of RNA found encoded by RNY and extended genetic codes? We have of all these types of RNA encoded by all of those genetic codes before LUCA. We have, for example, the RNA moiety of the RNase P that serves for maturation of the RNAs. We have also the RNA moiety of the signal recognition particle that carries the proteins to the protein complex to membrane. We have also the RNA 6S that is only bacterial, the tmRNA that is a dual molecule that mimics the translation termination complex. They are only found in bacteria. We have also very interestingly, the tRNAs. We have the 20 types of tRNAs, the 20 isotypes of tRNAs encoded, fully encoded by each of the extended genetic codes. We have also the ribonucleic, the ribosomal RNAs. We have the 5S, the 16S, and the 23S. So, for example, we found that the tRNA for glycine with the anticodon GCC encoded by, for example, the extended one. And we have, we can observe, yeah?
Yeah, oh. Kind of past your time now. So if you can, 2 minutes or something, round it up.
Yeah, I hurry up. Yeah, thank you. I'm almost finished. So we can observe that the tRNA for glycine with this anticodon is practically totally formed by if we observe the extended 1 genetic code. For example, the tRNA for cysteine with this anticodon, we have is not totally formed, but almost formed with encoding by extended genetic 2. And we have, as I mentioned, the 16S RNA, the 5S, the 23S that, for example, we have indicate here the PTC, the PTC region. For example, we have the encoded by RNY or the SRAP on all of these RNAs. So the key message is that we delineate and analyze the evolution of the proteome and the RNAome is starting with Eigen and Schuster primeval genetic code, followed by the two evolutionary routes, extended genetic codes 1 and 2, that finally gave rise to the standard genetic code that pertains to Luca. So we have observed that this dichotomous algebraically and biologically pathway is valid, not only plausible, is not only algebraically sound, but also we have tried to prove this pathway using our model by reconstructing the proteins and the RNAs. So we can observe as I mentioned, the extended genetic codes in this timeline, and we could position these encodings as the period that Prosdocimi and the Farias named as the FUCA, the first universal common ancestor before the last universal common ancestor. So, as Theodosius Obsensky says, nothing in biology makes sense except in the light of evolution. So, nature's great is written in mathematics, as Galileo Galilei says. Thanks.